In previous video, we discussed about visual processing and structure of retina. In today's video, we will elaborate the structure of retina in little more detail, and we will try to understand the receptor and neural function of retina. In previous video, we already discussed that the retina is the light sensitive portion of eye and is composed of 10 distinct layers. There are 3 different types of light sensing cells which are present in these layers. These are cones, rods and photosensitive ganglion cells. Now cones, they are responsible for color vision and they are also responsible for vision in bright light. We call it photopic vision. The cone system, it has a much greater visual acuity. Next, the rods. They are responsible for black and white vision and they are extremely sensitive to light. And hence they are the receptors for night vision, also known as scotopic vision. The rod system, it is incapable of resolving the details and boundaries of objects and even their colors. Each of these two systems, that is rod system and cone system, they work maximally under different conditions of illumination. This is called as duplicity theory. Next is photosensitive ganglion cell. These cells, they are important for entrainment of circadian rhythms and for reflexive responses such as pupillary light reflex. The peculiar thing about vertebrate retina is it is inverted. Now, what does it mean by inverted? We are saying it as inverted because the light sensing cells or photoreceptors, they are present in the back of the retina. So the light has to pass through layers of neurons and capillaries before it reaches the rods and cones. The ganglion cells whose axons form the optic nerve are at the front of retina. Therefore, the optic nerve must cross through the retina while going to the brain. So in the region where the optic nerve crosses the retina, there are no photoreceptors, which is indeed giving rise to blind spot. In contrast, in the cephalopod retina, the photoreceptors are in the front with processing neurons and capillaries behind them. Because of this fact, cephalopods do not have a blind spot. At first glance, one may think that the vertebrate retina is wired wrongly or badly designed, etc., etc. But the fact is that it is indeed perfectly designed by nature. The retina could not function if it were not inverted. The photoreceptor layer, it must be embedded in the retinal pigment epithelium for the retina to function properly. There are various reasons why retina could not function if it were not inverted and embedded in retinal pigment epithelium. So one most important reason is that the supply of oxygen and other necessary nutrients needed for the photoreceptors to function, they come from pigment epithelium. And this retinal pigment epithelium, it in fact extracts those nutrients from the choroid. Another reason is that when light strikes 11-cis retinal, which is a pigment which is present in the rods and cones, this 11-cis retinal, it changes to all transretinal, which then triggers the changes in opsins. Now, the outer segment do not regenerate the retinal back into the cis form once it has been changed by light. Instead, the retinal is pumped out to the surrounding pigment epithelium where it is regenerated and then transported back into the outer segments of photoreceptors. This recycling function of retinal pigment epithelium, it protects the photoreceptors against photooxidative damage and thereby prolongs the life of these photoreceptors. Now, one might think that as retina is inverted and light has to pass through all the layers of retina to stimulate photoreceptors, ideally the image should get blurred and thereby lose the visual acuity. But we all know that this does not happen. and our vision is in fact many times better than the image created by most expensive camera. Do you know how the nature has addressed this problem? In human beings and many other vertebrates, there is a small central portion of retina which is adapted for high visual acuity. We call it Phobia Centralis. This area, it is avascular, and in this area the other retinal layers are displaced to the sides, thereby leaving very minimal neural tissue in front of photoreceptors and hence very minimal light scattering, and thus responsible for providing us with crystal clear image of external world. Wow, amazing. So thanks to Phobia Centralis, because of which you are able to view my video and because of which I am able to create this video. Some more interesting points about Phobia Centralis are, it is only 1.5 millimeter in diameter, and yet half of the fibers in the optic nerve, they come from this area, and remaining half comes from rest of the retina. Phobia is also very rich in cones. There is a more smaller central portion of Phobia Centralis which we call foveola. This area is having only cones and no rods. Now the 10 layers of retina, they can be grouped into 4 main processing stages. First is phototransduction, then transmission to the bipolar cells, then transmission to ganglion cells, and finally transmission along optic nerve to finally reach the primary visual area in occipital cortex. Now let's come to phototransduction. So what is phototransduction? It is a process by which light is converted into electrical signals in the rod cells, cone cells, and photosensitive ganglion cells of retina. Retina contains about 7 million cones and 75 to 150 million rods. These rods and cones, they follow a particular distribution pattern. Like the central retina, it predominantly contains cones, and the concentration decreases as one approaches peripheral part of retina. Similarly, the peripheral retina predominantly contains rods, and their concentration decreases as one approaches center, and there are no rods in the foveola. Now different cells of retina, they basically release one of the two different neurotransmitters. So photoreceptors, bipolar cells, and ganglion cells, they release glutamate, whereas amacrine cells and horizontal cells, they are inhibitory cells and they release GABA. All the electrical responses of photoreceptors and most of the other neural elements of retina, they are local graded potentials, and only the ganglion cells are capable of producing all or none action potentials. Another important point to note is that the responses of rods, cones, and horizontal cells, they are hyperpolarizing, whereas the responses of bipolar cells are either hyperpolarizing or depolarizing. And finally, the responses of amacrine cells, they are depolarizing. For now, just trust me and remember these points. Later on in next video, these concepts will be cleared while discussing about phototransduction and its steps in detail. Now finally, let's very briefly discuss about structure of rods and cones. Both rods and cones, they are composed of outer segment, inner segment, cell body, and synaptic terminal. The outer segments, they are modified cilia composed of regular stacks of flattened saccules or membranous discs. The light sensitive photochemical, it is present in the outer segment. These cells, they also contain basal bodies and organelles like mitochondria and endoplasmic reticulum, and their cell body contains nucleus. Now the photochemical in rods, it is rhodopsin, whereas cones contains one of the three different types of photochemicals. They are porphyropsin, iodopsin and cyanopsin. The color which our brain perceives, it depends on which particular photochemical is splitting when the light strikes. Our brain perceives color as red when porphyropsin is split, green if iodopsin is split, and blue if cyanopsin is split. So that's all for today. In next video, we will discuss about phototransduction and its steps in detail. So don't miss my upcoming video. Thank you. Have a nice day.